Hello viewers and learners, welcome to this session. Today we will discuss on Epigenetics and Pollution Part 2, and this forms a part of the program Postgraduate Diploma in Environmental and Occupational Health and Master of Arts in Environmental and Occupational Health, and this will form a part of the course in the program that has a course code of MEV 004 Environmental Toxicology. Now to discuss this, we have with us in our studio Dr. Suresh Kumar, Scientist F from the National Institute of Cancer Prevention and Research Center Control from the ICMR located in Noida in New Delhi. Welcome to this session and to IGNOU, sir. And in the previous session, sir discussed about the methylation patterns and how we have seen some of the benefits of taking like the folic acid during some of the women who were in their first trimester, and how the iron or the folic acid is important for preventing the neural defects among the babies that were born. So that was some positive outcome of this DNA methylation and the processes involved. And there can also be pollution related problems which can alter this DNA methylation. So I just had one question, sir, that we have seen some of the benefits. So just like this, pollution can have an effect, and if there is some change which is observed, can that be reversible? I mean, can this be reversed back to its original state?
Right. So we can reverse it. May I can go to some more epigenetic mechanism that also have a role.
Yeah, of course.
Then I can answer your question.
Definitely.
So we can go for that other mechanisms which we have. Last time we seen DNA methylation as one of the mechanism that happens. So not a DNA methylation alone that do gene expression regulations. It also takes the help of histone modulations.
That's right. Okay.
So I'll come to this histone modulations. So basically, histones are a nuclear protein that is wrapped by DNA, and they are found as a nucleosome in the nucleus. So in histones, you have H2A, H2B, H3, and H4. These four histones, they found as a dimers. So these dimers, these octamers are wrapped by 146 base pair of DNA. So these histones are subject to variety of post-translation modification like any other proteins. The proteins, once it has been translated, they do undergo a post-translational modifications. So the post-translational modifications can be phosphorylation. So most of the kinases present in the cells, they get phosphorylated for its activation. And phosphorylation, acetylation, methylation, and ubiquitination. So ubiquitination generally that is been found in protein degradations. So these post-translational modifications that alters a gene expressions. This is also a epigenetic modification that happens in the histones. We have seen epigenetic modification in the DNA. In the cytosines, the methylation happens. Now we are going to see the modification that happens in histones, particularly in the amino acids. Particular amino acids in this particular histones, they do get modulated by acetylation or methylation. These two things which we are interested with our concerned with our epigenetic study. So as I seen that again, I'll just recapitulate the things that epigenetic mechanism, DNA methylation, histone modification, and non-coding RNA. These three all together, either independently, coordinately, they do gene expression regulations. So as I say that H2A, H2B, H3, H4, they are all found as a dimers. They do have wrapped around by DNA, and this particularly if you see that histone tails here, and particularly H3 histone tails here, this H3 histone basically is amenable for the modifications, either acetylation or methylation. When methylation happens, the particular H3, I mean histone 3, H3 dimethylation, trimethylation in particular amino acid, that is we call H3K4, that is H3K4 trimethylation. That determines the expression. When you have this methylation, this is a transcriptionally active methylation pattern. When this trimethylation become dimethylation, it become repressive. Same way, H3K27 methylation is a repressive mark. So the methylation in K4, trimethylation K4 is basically active form, and H3K27 methylation is the repressive mark. So methylation mark here play a different role based on the trimethylation, dimethylation in the histones, and also acetylation. Acetylation also basically, if you see that acetylation gives negative charge to the DNA. So when the acetyl group is added into the any of the amino acid, particularly lysine again, this amino acid gives attains a negative charge, and net negative charge attained by histones. When histone become negative charge and DNA also negative charge, they repel each other. So acetylation basically induces active gene expression environment. So if the particular histone is acetylated, so that genes which are wrapped around by histones, they become active transcriptally, right? And if you remove the acetylation, it become clumsy, that become tightly coiled. So the euchromatin state become heterochromatin state by removing the acetylation. So this is what you have a different modulations, particularly by acetylation, methylation. This K4, this is a K4 methylation, and K27 methylation is what important in our most of the studies which are related to the environment, pollution, as well cancer biology. So methylation in K27 is being found in most of the repressed state of genes, and methylation in K4H3 region, that is active region of genes. And this is actually, this methylation mark, all other methylation marks are been recognized by set of readers, writers, and erasers of methylases, basically histone methylases. So certain histone methylases they reads, the certain methylases they recognizes, certain methylases they erases. So this is a kind of a dynamic part of inclusion and erasure of DNA histone modulations. So organization of epigenome is like that. When this H3K4 trimethylation happens, these are basically transcriptionally accepted region, and when it is H3K27 methylated, it become heterochromatin. This is not transcriptionally active region. This is suppressive state. So this way, DNA methylation and histone modulation acting together, they do transcriptional suppressions. So this is a mechanism which coming to this part particularly. If you see that this is a gene which is having the second mechanism what I'm seeing here. This is a gene at the promoter region. You have transcription factor binding site is here. When it is methylated, it invites some of the molecule, and this molecule basically MECP2. This is basically suppressive complex, along with some of the histone deacetylases comes here. That actually suppresses gene expression. So this is just how the histones also, histone demethylases have a role in gene silencing, which I am telling. So basically relaxed transcriptionally active DNA is called euchromatin, and more condensed is heterochromatin. Here we see that more condensed, this is what more condensed region, this is what heterochromatin, and this is relaxed is euchromatin. So then I say that acetylation removes the positive charge on the histones, result in repulsion with the negatively charged phosphate groups of DNA. As a consequence, chromatin is transformed into a more relaxed structure that is associated with the greater level of gene transcription. So there are histone deacetylases and histone acetylases. So they do act in different direction, and so condensation can be brought by deacetylating the histones. So that way the regulation of gene expression has been monitored by the histone modifying enzymes. So this is how it happens. So in the normal cells, particular tumor suppressor genes is in a euchromatic state, and when this has been when this particular when this acetyl group is removed in the cancer cells, it becomes heterochromatized. So it becomes a repressive complex. So this is how the histone modulation they have role. So this is another example gene which is in a achromatic state and become heterochromatic state because of methylation and in conjunction with your histone modulations. So coming to your question, madam. So you asked a question, what is the good effect of I mean, we have seen in the DNA methylation the good effect of this thing. The bad effect, we'll see that what are this DNA methylases enzymes? It's like we saw that DNA methyl transferase is one, and histone methylases, histone demethylases. These actually there is a set role to maintain this methylation pattern, and it is inherited from mother cell to new daughter cells. This is exactly same copy is being made. If any alteration in this pattern is again fixed in the alteration, when the alteration happens in this methylation pattern or histone modifications, it is again fixed in the cells. So how we can avoid this? Now what we are now, there are a lot of now the pollution. The pollution actually causes the defects in methylation machinery. It interacts with the DNA methyl transferase expression, or it intercalates with the DNA strands, the pollutants. So there are a lot of mechanisms which actually alters the enzymatic activity of this methyl group also. So the recent technologies which is coming up, epigenomic studies, they are bringing a lot of evidences, and which we can make that this particular epigenetic alteration can be very well managed by advisories, formulation of advisories, and also lifestyle factors and dietary patterns, how you follow it up. So these things are coming up now, and with a new advent of technologies. Maybe I'll discuss in coming session also, coming about when we talk about the pollution, I'll discuss about those things. So third mechanism is
it is possible to be reversed.
Yes, that's what this methylation patterns are reversed, as we saw that when the folic acid, by giving folic acid, we could ensure that neural development is ensured. So same way we can reverse certain epigenetic patterns by management of by diet factors. I'll discuss about that. So the non-coding RNA, basically these RNAs are basically, they don't code for any protein. So they do function in some cellular physiology, but they don't do any functions. So one is that housekeeping RNAs, which we call tRNA, rRNA. These are the one which are normally functioning in translation as well, translation process, protein synthesis process. There is another class of RNAs which are classified based on the size. One is small RNA and long non-coding RNAs. This small RNA is basically they transcribed into the small transcript and processed into smaller, like 21 bases of RNAs. These are called miRNAs, siRNAs and all. And long non-coding RNA can be more than 200 bases. That is a hot air RNA, telomerase RNA. These are the example for non-coding RNA, which has functional in the cellular physiology, which has a function in cellular physiology. It can act as if you see that the particular long non-coding RNA, they can help in splicing modulations, they can help in, they can regulate the enzyme, they can act as a sponge in the sense that it can regulate the miRNA expression, they can also help in translocating protein to a particular site. So it does a lot of functions. So long non-coding RNAs, small RNAs, they do have epigenetic functions. So the presence or absence of this RNA, they have an effect. The presence and absence of this non-coding RNA is mediated by, again, DNA methylation and histone modulations. So now the pollution, coming to the pollution, how it is relevant to the epigenetics. The pollution is defined as any undesirable change in physical, chemical, biological characteristics of air, water and land that may be harmful to our living organisms. So there are different pollutants. There are primary pollutant, secondary pollutant. Primary pollutant, we generally directly phase the primary pollutants, and based on the nature, you have solid, liquid, gases, different types of pollutants. And based on decomposition, you can say biodegradable and non-biodegradable. So the epigenome players, basically the proteins which play a role in acetylation, methylation chemicals, they become sensitive to this environmental cues. They become sensitive to this environmental stresses and they get altered. The methylation machineries, the enzymes, the methyl transferases and DNMTs, they get altered because of this particular pollutions. How they get altered, we'll come to see. So there are changes that have important effect in the phenotype of the organisms. They induce the pollutants. They not only alters the phenotype of the cell, it induces changes in DNA methylation, histone modification, and also non-coding RNA expression. So generally it alters the epigenetic status of the cells. If you see that this is the chromosome, so you have P arm and Q arm, and this is so this is a telomere at both the ends, and this is a centromere. And these stars which I have written, that this is basically depicting a methylation pattern. So you have, in normal cells, you have hypermethylation and whole genome. Hypermethylation, you have a lot of stars here, that means a lot of methyl cytosine content. Okay, so in the normal cells, you will have a lot of methyl cytosine content, whereas the exposure to pollutants reduces this number of methyl contents, in the sense that the hypermethylation become hypomethylation, right? The number of methylated cytosine reduced here. So number of methyl cytosine reduced here means it is hypomethylation. As you know that there are certain genes are kept off in the normal cells. For example, like oncogenes, proto-oncogenes, like c-MYC. There are other genes called transposons. These transposons we acquire through viral infections. These transposons are kept silent by methylation process. But this pollutions, what it does is that due to hypomethylation, hyper to hypomethylation, the genes which are silenced by methylation, they become active. The c-MYC, it is basically a proto-oncogene. They become active. So these oncogenes basically they helps to cells to proliferate more. Same way that the transposons. Once the transposons get active, they started jumping one place to another place in the genome and creates a genomic instability. So there are multiple jumping genes present in the genome. That means that it's not good for health of the genome. So that way it creates a genomic instability in the cell. So hypermethylation, so here that we see that hypermethylation goes into hypomethylation in the chromosomes. So and also you see that
these environmental changes, like whatever you said now, so they can also be managed in the clinical setup?
Yes, this can be managed very much in clinical setup. I'll give you some examples in the coming slides. Maybe some of the examples in the slides. There are a lot of studies have been done. So when the particular pollutants has been exposed to the cells, they do methylation in particular genes, and they have in few studies they have managed it. That I'll tell you.
And also I have some more interesting questions, but according to the thing you can see, like these days, like in olden days we were not exposed to so much of the pollutants, even from the cosmetics or whatever we were using, because we were only using maybe the handmade soaps or the powders or the whatever concoctions that we were getting from the trees or the bark of the trees. But nowadays when you go for a shampoo or a dye product or anything, just flip the label and you find so many of the chemicals in it.
Right. Yes.
So does this also have an effect on?
Yes.
And then still, why are they being sold, and why are people still wanting to buy them off those?
So this is what the
shelves.
strategy, what you said, that how we can manage clinically or how we can manage it. This is for exposure reduction measures we have to take. It's personally as well government policies like exposure reduction.
Because most of the people will not be knowing this.
Right.
You want to flaunt nice hair.
Right.
You just go for the product.
Exactly.
So they are not able to explain.
Right.
And I often go and I ask them that there are a list of the chemicals and still you're asking me to buy. So where is the logic in this?
Right.
So does that also have an effect on the
exactly.
DNA?
It has an effect, yes. Okay. All the things which is, whatever the abnormal things, whatever we have against nature, that all have, that alters the epigenetic. And whenever using, there is a Short-term usage creates some short-term usage creates a hypermethylation. Some of the chemicals, when you do chronically, they have a different methylation patterns that we can see that. So as we see here in this particular slide, we have a hypomethylation for hypermethylation to hypomethylation in whole genome that happens. Yes, this is what the slide. So here, but if you see the regional hypermethylation happens, the regional hypermethylation happens here. Certain genes are heavily methylated, whereas this is not so. Only particular region is hypermethylated. So genome hypomethylation and regional hypermethylation that happens in the cancers, particularly when it is exposed to certain pollutants. So there is a regional hypomethylation in the normal, and whereas the cancers, it is regional hypermethylation in the cancers. So we know that, as I said, that I'll come to your point by toxins. I'm just telling about toxins. So we see that arsenic is heavily contaminated. I mean, arsenic toxin we do find nowadays in the water, contaminated drinking waters. So the arsenic, basically even Gangetic plain, the arsenic contamination has been found. The arsenic, what it does is that when we ingest arsenic contaminated water, they get detoxified. So when this arsenic during the process of detoxification, they need this S-adenosyl methionine again for biotransformation for its detoxification. So what it does, it tries to take up all the SAM, S-adenosyl methionine, and it gets transformed into multiple forms. When SAM is been taken out from the cellular pool, the cell become deficient of SAM pool. That methyl donor pool is been deficient. So what happens is, obviously the normal cellular process which you need to have hypermethylation, the genomic hypermethylation, that become hypomethylated. So this way arsenic extracts all the pool and it induces hypomethylation in the cells. Same way cadmium shorter exposure, it induces particular gene, suppresses a particular gene expression like hydroxysteroid beta hydrogenases 2. And same way cadmium, a longer exposure that elicits a hypermethylation of P15, P16 genes. This P15, P16 genes are cell cycle regulated genes. These are basically the genes which acts in the initiation of carcinogenesis process. When this P15, P16 are suppressed, then the cells are prone for further epigenetic insults that may lead to carcinogenesis process.
So this may also be one of the reasons that these days we are finding a lot of the carcinogenic and cancer cases.
Right.
Because the water is totally contaminated and you find a lot of heavy metals, industrial pollutants.
And heavy metals, not only the abstracts as SAM, it also induces reactive oxygen species once it's get into the cells. So it generates a ROS. When it has a reactive oxygen species enters in the cells, I mean not enters, it's created in the cells. So it induces a DNA damage. So when they induce a DNA damage, that means it also induces a mutation in the cells. So mutation and methylation coordinately helps in carcinogenesis process. So it has a lot of effects, this metal exposures. So mercury exposure also the same way, nickel exposure is the same way. So there is another pollutant called persistent organic pollutant, which is something stays in the earth for more time. It is more than 60 days, it is not degradable. So these are basically comes from pesticide storage or agriculture fields, everything it comes back. And when it comes to the animals, when animals eat some of the grains and grasses, these animals, when human consumes, all the pesticide comes to the human body. So the human body accumulates this pesticide, which is not degradable. So they also create modulations in histones and expression and this thing. So there are around 19 chemicals that have been called as an POPs, but now government has been telling that it's more than 200 things comes into the as a POP. So we don't have list. A lot of lists are which can create epigenetic changes in that. So for your question, so one of this is one of the study which they have taken that the Bisphenol A, which is
Bisphenol A is
Bisphenol A
plastic,
present in the plastic bottles,
bottles.
water bottles, even
Feeding bottles,
bottles
babies,
particularly, yes. Bisphenol A has been noticed in this water bottles and feeding bottles. And they have done an experiment with the animal models in which that bisphenol A ingested animals, generally normally they develop a brown color coatings. When bisphenol A consumed rats, I mean mouse models, they formed a yellow color coats. So when they found out why this is happening, they found out the color gene which is called agouti gene, that the upstream of agouti gene has some transposons, they get activated because of bisphenol A. So because of hypomethylation of transposons, they get activated, and that actually changes the color code of your this thing. So for your questions, when we have this many bisphenol A has been consumed in the same mouse model study, they supplemented this mouse with folic acid. This mouse has been supplemented with the folic acid, and this folic acid supplementations then nullify the effect of BPA.
Okay.
Right. So the mouse model which has been a yellow color, it become brown color back, or the brown color is not transformed to yellow color. So they found that supplementation of folic acid has a role in beneficiary effect in altering DNA methylation. So like many of these metals have a different, so the arsenic has global methylation, they have a regional methylation P16, there is a death associated protein kinase methylation. So many genes are methylated by arsenic and many genes are hypomethylated. So many environmental chemicals, they do alteration in the genes. These are the list, there's a growing list that alters DNA methylation as well as histone modulations. So also miRNA, also like aluminum, they do induce some miRNAs which are basically not good for health, and also mercury also they does some hypomethylation. So every pollutant, they do alters methylation pattern of normal methylation pattern of the cells, and it indirectly helps in developing the same. So in summary, I'll say that so alternation in DNA methylation provides clue for identifying the pollutant based alternation methylation, and also physiological consequence that we can identify by assessing the DNA methylation. And also this alteration, they do alter the DNA methylation and many of these histone modulations, and there are P16, P15 methylation happens in the cells, which shows that these are the cell cycle control. This itself shows that when there is a pollutant affects this particular genes, we have to be a little bit careful that these are the genes which are cell cycle regulatory genes. When these are shut off, the cells keep going on in their cellular progression. So we have to take care of this thing. And as we say that this can be reversed by adopting lifestyle dietary factors. So one more example which I can give is that since we are living in a heavily contaminated, air polluted areas like in Delhi, we have that the particulate matters, 10 microns and 2.5 micron particulate matters. The 2.5 particulate matters create a lot of epigenetic defects. It induces immune deficiency. It alters a lot of epigenetic machineries. So P25 and this 2.5 micron particulate matter. So one of the study from Columbia University in 2013, I hope. What they did is that they have experimentally exposed the human with the particulate matter for 10 days. Within 3 days, they do find changes, epigenetic changes. In 3 days, they found epigenetic changes. So they go through the 10 days of exposure of this 2.5 micron particulate matter. So the humans exposed in a control environment with this, then they have given supplements. They gave vitamin B. So when the vitamin B was given, the epigenetic alteration that has been induced by pollution has been reversed. So the important observation is that we have to reduce our exposure reduction strategies. We have to develop exposure reduction strategies. We have to adopt a lifestyle in which that it is necessary to keep our health important by consuming healthy diets which has all these vitamins, folic acid supplements, vitamin B, C, folic acids, B12. They all help in ensuring your normal methylation pattern is setting up in the normal status in the cells. So I think with that, if you have any questions, you can. Thank you.
Thank you, sir, for explaining like the epigenetics. And in this session, we actually discussed more about the effects of environmental pollution on the DNA, and you gave a lot of examples about the heavy metals in particularly the arsenic, then cadmium, and also mercury. And also another important thing that was bisphenol A, which is also found in many of the plastic bottles. And so that is why people are now turning to even the metal type of the bottles. But again, though the silicone nipples that are found on the bottles also, there are several issues. So there are environmental pollutants at every step. There is so much to be discussed. But as you say, maybe there could be the problems can be diagnosed, and we need to come up with eco-friendly solutions. And one thing is, yes, these dietary patterns and taking of these B complex and folic acid, which of course under the supervision of medical practitioner, we need to take them in the right dose, and that will definitely have a very good effect on our health and the living systems. So thank you, sir, for explaining all the concepts. And my dear learners, I hope you have had a good understanding of this subject. Thank you for your patient listening. Thank you.
Thank you.